Hello viewers, welcome again to our ongoing series on the mechanism of photosynthesis. Moving forward towards this journey, you remember learners, in our last lecture we had talked about the details of the popular Calvin cycle or the so-called C3 cycle. Now this cycle became universal and even Professor Melvin Calvin received a Nobel Prize. Now what transpired a little later was very interesting, because people thought that this was perhaps the universal pathway in all the plants and the photosynthetic autotrophs, that is the photosynthetic bacteria. But then something else was in store. So we should first remember that the C3 cycle is in fact a universal cycle, no doubt that it utilizes the Calvin enzymes. And what really happened was that as this oxynogenic photosynthesis proceeded in the world, there was a gradual reduction in the carbon dioxide levels, which were initially very high, and oxygen started building up. Now this definitely, a certain threshold level would have come say around 350 years earlier. Now when this whole so-called oxygen revolution came into existence, this must have triggered a series of adaptations, because now oxygen was a bit higher and this led to the problem of photorespiration. You remember learners, we had a detailed discussion on the so-called C2 cycle or the glycolic acid pathway, nicknamed as photorespiration. Now there must have been several selection pressures in view of the enhanced oxygen concentration, which must have really engineered the evolution of certain alternative pathways which could be more efficient. More efficient for what? For number one, release of carbon dioxide in the immediate vicinity of rubisco. Because if you remember, we said that rubisco is a bifunctional enzyme. It could act as a carboxylase as well as an oxygenase, depending upon which of the substrates is in higher concentration. So there was one strategy. The other one was to avoid the use of ATP and the reductant which was required to recover the ammonium ions. So nature must have adopted various strategies to very efficiently uptake carbon dioxide and the bicarbonate ions from the surrounding environment. And somehow if these could be concentrated and channelized near the rubisco, then of course the concentration of carbon dioxide would have always been higher than oxygen and the carboxylase activity would work. But then we find that this type of an oxygenation reaction, if somehow can be avoided, photorespiration be reduced, naturally it would result in an increased productivity among the C3 plants. So in other words, the natural selection or the evolutionary natural selection machinery would have really evolved certain CO2 concentrating mechanisms so that in order to have the induction of more and more of improvement in carboxylation efficiency. Two types of mechanisms perhaps come to our mind which would have taken place. Number one, carbon dioxide and bicarbonate pumps at the plasma membrane of certain photosynthetic bacteria, the cyanobacteria, blue-green algae, that is eukaryotic algae, and various aquatic plants. And then some of the biochemical mechanisms. That means why not synthesize a 4C compound first before it reaches the Bensel-Calvin cycle, and through this intermediate product concentrate the carbon dioxide near the Calvin cycle so as to efficiently run and avoid somehow the participation of oxygenase activity. Now, these two are the subjects of today's discussion, and that is the Hatch-and-Slack cycle or C4 and the crassulacean acid metabolism, which we nickname as CAM plants. But learners, you should appreciate one point. When the carbon of carbon dioxide is in the atmosphere, can you imagine so many barriers it has to pass through before it actually reaches the chloroplast stroma so as to get fixed in the Calvin cycle? Naturally, it has to pass through the cell wall, the plasma membrane of the cell cytoplasm, and then when it reaches the organelle, even the chloroplast envelope, it has to pass. Although there are various aquaporins which are going to decrease the resistance of mesophyll, and thereby they will facilitate the diffusion of carbon dioxide, yet there is a whole lot of series of barriers which are present. The story of C4 pathway is very interesting. There was a Russian scientist by the name Korshak and Karpilov. They were working on, in fact, the C3 cycle, and they wanted to repeat this Benson-Calvin cycle in certain monocots like sugarcane and maize. So they provided radioactive carbon, that is C14O2, to sugarcane. To their utmost surprise, they found that the label of the carbon was not in the form of a 3-carbon compound, that is PGA, as we saw in Calvin cycle. It was in fact in the form of a 4C acid, that is malic acid and aspartic acid. Now, this was very, very surprising. So, that means they said there must be a different type of a pathway which is being operative in these monocots. And later the details were mopped up and found out in details by 2 young Australian scientists, Hatch and Slack, and who elaborated and elucidated the entire pathway, and therefore it is called as Hatch and Slack pathway, or more appropriately as Hatch, Slack and Korshak pathway, that is the HSK pathway. As you can see on your screens, sugarcanes is a very important example of C4 pathway, and so is the corn or Zea maize. Of course, this type of cycle also takes place in tropical grasses. Actually, it is a alternative carbon dioxide fixation cycle, and since the first products are not C3, they are in fact C4. So, it is more appropriate to call this cycle as a C4 cycle or a dicarboxylic acid pathway. Now, this is a strategy which is directed towards concentration of carbon and somehow transporting this carbon in a concentrated manner to rubisco in order to minimize the oxygenase activity of this bifunctional enzyme. And on one hand, photorespiration associated energy losses would be minimized, but then very soon we would see that every such additional activity comes with a cost, and therefore this may or may not be a very energy efficient process. So, what are the unique features of C4 plants? It concentrates the carbon dioxide by initially fixing it by a new set of enzymes which is headed by PEP carboxylase, which is also called popularly as pepcase, phosphoenol pyruvate carboxylase. Now, this enzyme which has been evolved has a very, very high affinity for carbon dioxide. It can even trap CO2 at minimum level of 2 parts per million. You remember the enzyme rubisco, its threshold would be only 50 ppm, and it would start becoming or functioning as a oxygenase below that. So, there is a spatial separation of PEP carboxylase and rubisco. That means there are 2 different tissues in which these 2 reactions involving these 2 enzymes are operative. And for this, there has been a very apt evolution of compartmentalization with a very specialized morphology called as Kranz anatomy. If you see, learners, on your screen, what is a typical leaf anatomy of a dicot leaf, and that is of a C3 plant, there is a simple palisade parenchyma and spongy parenchyma, and then in the middle you find a vascular bundle with no additional structures. But when we compare this to the Kranz anatomy, we find that there is a ring of mesophyll cells which is surrounding the cells of the bundle sheath. The bundle sheath cells are tightly packed, and this whole structure gives us the appearance of a wreath. I am sure you know what a wreath is. On a departed soul, we always put a floral bouquet which is round, circular, and that is called as a wreath. So, in German, wreath is called as Kranz, and hence the name Kranz anatomy. Incidentally, this structure was first observed by none other than the famous German biologist Haberlandt in 1884, and he said that if there is such type of an anatomy, definitely there must be some division of labor between the mesophyll cells and the bundle sheath cells. So, that means they must be performing different functions, and his prophecy came true, because if you look at the anatomy of C4 leaf, even the plastids are of two types. We call them as dimorphic plastids. We have never mentioned two types of plastids in C3 plants. So, it is very interesting to note that the plastids of the mesophyll cells are small, but then they possess grana. So, they are called as granule, and the plastids which are present in the bundle sheath are agranule, that is, the thylakoids are not arranged in the form of grana, and these particular plastids are much bigger in size, and they are having much abundant starch. So, if they have starch, that means maybe the sugars are being produced in the plastids of the bundle sheath, and both these plastids, they are connected by plasmodesmata. If you were to look on your screens at a very beautiful electron micrograph of the two types of plastids which we call as the dimorphic plastids, we will find that we have the granule plastids in the mesophyll and the agranule ones in the bundle sheath. And of course, you can also observe the plasmodesmatal connections between the two. So, that means there is a distinct labor, division of labor between these cells. How? We find that the non-cyclic light dependent reactions that make oxygen, ATP and NADPH, that means the light reaction, it takes place in the plastids of the mesophyll, and the cells of the mesophyll do not perform the Calvin cycle, because we find that PEP carboxylase is present and not rubisco. So, that means some other mechanism is going on, whereas the cells of the bundle sheath, they perform cyclic light dependent reactions. That means NADPH2 is not going to be produced, and a lot of ATP is generated, but then no oxygen is generated, and the usual C3 cycle is being performed. The cells of the mesophyll show a different cycle, which is the Hatch and Slack pathway, and the cells of the bundle sheath, they show the Calvin cycle or the C3 cycle. On your screens, you can find a pictorial depiction of a beautiful differentiation of the bundle sheath cells in a 3D manner, which are being surrounded like a wreath by the mesophyll cells. And this is how the cycle proceeds. In the mesophyll cells, where you have an abundance of PEP carboxylase and the abundance of Calvin cycle in cells of the bundle sheath. If we were to look at the entire process stepwise, there seem to be 5 distinct stages in the C4 cycle. Number 1, in the mesophyll, the story begins because something additional has to take place in addition to the conventional Calvin cycle in order to concentrate. In other words, more and more CO2 in a concentrated manner has to be fetched, trapped and sent or channelized to Rubisco in the cells of the bundle sheath. So, the first step would be the CO2 is trapped as bicarbonate ions, and as I said before, PP carboxylase has a very high affinity. So, that means it traps the bicarbonate ions, immediately it forms oxaloacetate and later malate, which are both 4 carbon compounds, and hence the name C4 cycle. Now, these 4C or the 4 carbon compounds have to be transported through the plasmodesmata to cells of the bundle sheath. Once they come into the bundle sheath, then there is again a decarboxylation of this 4 carbon compound, that is malate, into pyruvate, and carbon dioxide is released. So, that means CO2 was fixed once in cells of the mesophyll, and now is the time for second fixation of CO2 into the C3 cycle or Calvin cycle. So, actually it is a 2-in-1 pathway, and then finally pyruvate is transferred back into the mesophyll cells in order to get converted. So, that means in the mesophyll cells the pyruvate now gets converted into PEP, which is again the substrate. So, if you were to look at a summary diagram of C4 cycle, we find these different pathways which are being operative. And if you were to see in details the different steps, we find that the initial substrate, as I said before, is not carbon dioxide, it is bicarbonate ions. So, definitely when the bicarbonate ion is to be fixed, then there is an enzyme which is called as carbonic anhydrase. You remember, carbonic anhydrase is the fastest enzyme with the fastest turnover of about 36 million, and it happens to be present in our blood also. And this enzyme is a zinc containing metalloenzyme, and if you were to calculate it otherwise, it would one molecule would catalyze roughly 106 reactions per second. And then this particular enzyme now is going to supply CO2 to phosphoenol pyruvate in the C4 plants, and then finally to the Rubisco. So, that means PEP is not sensitive to oxygen. This is a very important evolutionary advancement, because Rubisco is highly sensitive, but pepcase is not. So, on the other hand, it shows a very high affinity for bicarbonate ions, and therefore, it can fix carbon dioxide many more times than Rubisco. So, once OAA is formed, it gets reduced to malate by the enzyme malate dehydrogenase, and in this process the coenzyme 2, that is NADPH plus H plus, they are used. Now, malate once formed is now going to enter with the help of plasmodesmata into the bundle sheath cells. Once it goes into the bundle sheath cells, there it gets decarboxylated, because our ultimate aim is to get carbon dioxide, which the cell was not able to receive because of the anatomy, because of such high temperature and light conditions prevailing for the C4 plants in the tropical grasses or in the tropical climates. So, therefore, this interior location of the cells of the bundle sheath could get CO2 through the Hatch and Slack additional cycle. Now, NADP here gets reduced to either NADP plus by the malic enzyme, or it gives rise to NADPH plus H plus. Now, the labeling experiments have shown some very interesting aspects, because as I said, CO2 is being fixed twice. So, the CO2 molecule which is released in the bundle sheath cells is the same which was fixed into the OAA in the mesophyll cells. So, it is the same CO2 which goes. Now, the CO2 is fixed now for the second time by Rubisco in the C3 cycle, and finally, the pyruvate gets transferred from bundle sheath back into the mesophyll cells. But when it is going back, then there is a case of simultaneous phosphorylation of two molecules of pyruvate by ATP. And in this process, we find that an additional molecule of ATP is required to transform AMP to ADP. So, that means in a nutshell, two ATP molecules are lost, or they are utilized to keep this reaction going. So, that means there is an additional expenditure. So, if we were to look at the summary diagram as to what is going to happen in cells of the mesophyll, we find that the malate which was going back is now coming back as pyruvate, and this pyruvate again is going to be phosphorylated into phosphoenol pyruvate by an expenditure of 2 ATP, and the Kelvin cycle runs in cells of the bundle sheaths. So, if we were to look at the energy requirements, we find that the fixation of 6 molecules of carbon dioxide through this C4 pathway would require, say, 6 into 2, 12 ATP, and the usual 18 ATP are required for the Kelvin cycle. So, in total, the ATP requirement now is 12 plus 18, that is 30 ATP per one molecule of glucose synthesized. So, that means it is 18 in the C3 versus 30 in C4. At the face value in the first instance, it happens to be an expensive process, but then one should remember C4 pathway is not totally independent of Kelvin cycle. Actually, it is a supplement or considered to be as an adjunct of the C3 cycle. This is actually a CO2 concentrating mechanism, and ultimately PCR cycle has to run, that is C3 cycle has to run. So, as I said before, even though it is a costly process in terms of energy input, but then it comes with a price, because the rate of CO2 fixation is almost 2 or 3 times more, and the photorespiratory losses are much less. So, that means even if the cost is higher, it is worth it, because you are minimizing photorespiration, and such plants are located in hot and dry environments. Normal C3 photosynthesis will not work beyond a certain temperature and light intensity. So, that means the gain in a C4 cycle is much more than it compensates for the energy expense in terms of the C4 plants. So, these are the various advantages that we have, and in our next lecture we would now think in terms of the rationale as to why did this C4 cycle come into existence as far as the evolution is concerned. Then the second question in our minds is, who came first, C3 plants or the C4 plants, and then what would happen when the environments are very, very hot and dry, for example, in deserts. So, that would have led to the evolution of another type of a pathway, which we would call it as the crassulacean acid metabolism. Thank you.